Welcome everyone to our first Grand Rounds presentation in 2019. And I want to wish you all a Happy New Year. And thank you for coming out despite the weather. And I'm happy that now the days are starting to get longer, so we will have more light potentially soon as we start our Grand Round series. We're very excited to have Dr. Christopher Monk here today. But before I introduce him, I do also want to welcome Dr. Larry Wissow, who is also with us for the first time today in his role as chief of our division. Do you want to stand up in case there are people who haven't actually put a face to the name yet? And that I wanted to make sure people know that after the Grand Rounds, there's a reception that we're having to welcome him. So, but first, we will have our Grand Rounds presentation entitled Adversity, Resilience, and the Developing Brain, which really hits so many of the topics that we as a faculty have said that we want to focus on in our own educational journey and our career development, to really think about the role of neuroimaging and thinking about different regions of the brain and how they are relevant for different psychiatric illnesses, as well as thinking about diversity issues, including poverty and low SES and race, and how they may impact mental health issues. So Dr. Monk is coming here from University of Michigan, where he's a professor of psychology and also a professor in the Center for Human Growth. And he has a history here in Seattle. He grew up here and also got a bachelor's degree in psychology here. Then went on to get his PhD in psychology with a minor in neuroscience at the Institute of Child Development at University of Minnesota. And while there, he did his dissertation on electrophysiology and behavioral measurements, and also had a lot of interaction with people that we know, including Dr. Sarah Webb, who's here, both being in the same lab with Charles Nelson, and also, as I learned, having a period of time where they were housemates. He also did a postdoctoral fellowship at NIMH, where he worked with Dr. Danny Pine in the section on development and affective neuroscience and the mood and anxiety disorder program. And over the course of his career, he has really built a great body of work in looking at neuroimaging and its impact on mental health, including a variety of disorders, but with a real specialization on anxiety disorders, and is really doing what we have talked about many times in the Grand Round series as a goal of looking at the neuroimaging changes that occur over the course of treatment for youth with anxiety disorders. So I will let him tell you more about the amazing work that he's doing, but we very much welcome Dr. Monk today. Thank you.
Great, thank you for coming right after the winter holidays. It's a real treat for me to be here. I grew up a mile and a half from here, just over in View Ridge. I was growing up in the 70s and 80s is the time of real free range, free range child rearing, no bike helmets, things like that, and I had 3 visits here through the ER, and this is seriously the first time I've stepped foot in this hospital when I'm not sobbing or crying, so I feel I'm really ahead of the game already, and it's only 8:10 a.m. All right, so I have about 50 minutes, 50-minute talk, or give or take, and I'm happy to answer questions along the way that may come up. These are really new data, and I am still just trying to wrap my head around some of it. It's the data I'll be presenting, we're solid about it. It's not like we just analyzed this and we feel confident about it, but I'm still trying to understand it. So if you have questions, I'm more than happy to discuss it. Also, I am an experimental researcher. I'm not a clinician. I work with clinical psychologists and psychiatrists who are clinicians. I know many of you are clinicians, and so your insights into these data, from my standpoint, would be particularly appreciated. Okay. All right, so before I begin, I just want to disclose a financial interest. I actually have stock in Johnson & Johnson. I've never had to disclose that before because it's not a conflict with the research I do. I just happen to own this stock for a long time, so I was just asked to put that slide up. Okay, so let me give you an overview of what I'll be talking about over the next 45 minutes or so. So I'm going to give some background on poverty, stress, and mental health, and then I'm going to introduce this existing study that we've piggybacked onto called the Fragile Families and Child Wellbeing Study. I'll give you an overview of that, and then based on the background I gave you, I'll offer you a theoretical model that we've created hypotheses of, and then had those hypotheses pre-registered, so you know we're not just post hoc cherry picking the data. I'll provide that model to you, and then we will revisit it at the end. And then I'm going to present our data so far. There's a lot more analyses we have to do, but I'll present our initial findings that we've acquired over the last few years. I'll give you the demographics of this sample, looking at SES, rates of psychopathology, and I'm going to spend some time talking about our brain imaging findings, which I'm really excited about. And there are two, just to clarify ahead of time, there are two different methods that we use. We use fMRI, where we're looking at activation in this task, in this case, in response to a task. And then we're looking at, we also look at diffusion MRI, which allows us to look at white matter tracks in the brain and how they develop. And then at the end, I'll revisit our theoretical model, show you what hypotheses were confirmed, what have not been confirmed, and then provide you with a revised model based on that. Okay, so there are a lot of really nice epidemiological studies out there showing that growing up in poverty is associated with a host of adverse outcomes, such as mental disorders like anxiety and depression. But there's some real huge questions that still need to be addressed. One has to do with specificity. So what are the key variables of poverty? We know there are a whole host of variables that are associated with poverty, but we don't really know what the key ingredients are that lead to some of these specifically bad outcomes. And I just want to acknowledge Kate McLaughlin and Margaret Sheridan here. Kate was here at the University of Washington until very recently, and they've done a lot of thinking on this and trying to push the field forward in terms of thinking about specificity, and they have a threat deprivation model that we've altered a bit, but we worked off of, and so I just want to acknowledge that ahead of time. The other area that's really not clear is how do these variables that are associated with poverty, how do they get under the skin and alter brain development in such ways to lead to these negative outcomes, like these increased risks for anxiety and depression? We really don't have a handle on either the specificity or the brain aspect. So to try to answer these questions, we teamed up with this study that I mentioned a moment ago, the Fragile Families and Child Wellbeing Study. Does this look as white to you as it does to me? Yeah, sorry, it's very bright, but that's it. This is the United States, of course, and it shows the different cities where they have samples from this existing project. So the Fragile Families Study is a birth cohort study of children born between 1998 and 2000, and they're in 20 of these U.S. cities across the United States. And the study is nationally representative of children born in cities of 200,000 or more. So that's nice. We know who this sample generalizes to, which is unique, which is very unique for those of us who are interested in brain imaging. And it's also representative of children in each city as well. And I'm a developmental psychologist, and from a developmental psychologist point of view, the study is really also phenomenal because they've already assessed the children at birth, 1, 3, 5, 9, and then again at 15. So there's this really rich longitudinal data that's available to look at as well. Okay, so that's the existing Fragile Families study. The PI on that is Sarah McClanahan. And let me just talk about the study that we're doing that's piggybacking onto the Fragile Families study. And that is, it's a very nebulous term, the study of adolescent neural development, or SAND. And so what we're doing in this study is we're looking at a subset of the cities, the 3 closest cities to us. We started with Detroit and Toledo, which are between 50 minutes and an hour and a half away from the University of Michigan. But then we wanted to get more subjects, so then we branched out to Chicago, which is a bigger trip and requires a couple days stay for the families. It's about 4 hours away from the university. Okay, so we successfully recruited 237 of these families from this existing study to the University of Michigan. And they spent the whole day with us where we collected data. I highlighted what I'll be talking about today, particularly task-based fMRI, looking at brain activation, as well as diffusion, looking at white matter tracks, but we also collected structural MRI. In addition, I'll be talking about the mental health in terms of diagnoses. We also looked at a lot of measures of symptoms. We assessed maltreatment. We have looked at, again, I won't be talking about this, but we looked at HPA axis functions such as cortisol from saliva and cortisol from hair. And we also acquired genetic and epigenetic measures. And amazingly, we had this tremendous graduate student, Adrienne Woods, who took it upon herself to get as many of the school records as possible from the kids in Michigan, Ohio, and Illinois. And so she'll be looking at those data in the coming years. Okay, so this is our study. Oops, let's just get off. And this is a brutal slide. I don't want, people do not need to look at this. The whole point of this is just to show that thanks to this existing Fragile Families study, there's all this great data from 0 to 15 that we can work from. And then this is our present data collection. We collected the brain imaging data and all these other questionnaires and everything at around age 15. And then we're in the midst of this part doing the follow-up. 2 years later, they just answer questions via the internet or mail, whatever they prefer, with the idea that we can link these measures, in particular the brain imaging measures, to how they do 2 years later. Okay, this is an MRI. I'm sure everyone's familiar with what the MRI looks like. We use basically the same thing with functional MRI. It's just a bit bigger. And this is what the fMRI setup is like. I just want to show this to you for those of you who don't do this for a living. You have everything that goes in there has to be non-magnetic. So you have a button box here that they press, that's fiber optic cables, and we have, in this case, we have them do a task where they see emotional faces. That is presented here on the screen. And then they have these prism glasses that allow them to see beyond their chest to the screen where the images are being projected. Okay. Okay, I also want to very briefly, I'm not a biophysicist, but I want to very briefly give you an idea of what the fMRI signal is. It's called BOLD, or blood oxygen level dependent signal, what that is. So you have increased neural activation and in the brain, and what that does is it leads to an automatic increase in blood flow to that area. And you have too much, the result is that you have too much blood flow going to that area where there's a lot of neural activity. And so you have more oxygenated relative to deoxygenated hemoglobin in in that area, and this leads to changes in the magnetic properties in that local region of the brain. And so the idea here, and what I just want to acknowledge off the top, is that this is an indirect measure of neural activity. What we'd like to do someday is be able to look precisely at this neural activity live in the brain, but without surgery. That is not possible, so we're left with measures such as fMRI and other related measures that allow you to get that measure of blood flow. Okay. Okay, so let me give some background, some relevant background on brain function. I'm going to talk about two different circuits. One is the threat circuit, and then other one is the reward circuit that will be relevant for a theoretical model. So the amygdala is, for some reason, my favorite structure, apparently. If you look at my CV, a lot of the papers we write are related to the amygdala. And so the amygdala is part of a network that's very highly responsive to salient stimuli that are picked up in the environment, such as anything that may be of threat to an individual will activate the amygdala. And there's a ton of data on this. It's highly responsive to highly involved in detecting and recognizing emotional faces as well, and that emotional faces are certainly salient stimuli that a human experiences. Also, patients, both kids and adults with anxiety disorders, have greater activation in the amygdala to threatening stimuli, such as fearful faces and also angry faces. And beyond looking at disorders, it's also affected by the environment. And this is a really nice meta-analysis that a graduate student in my lab, Tyler Hine, that she did that was recently published. She looked at all these studies, 20 studies of maltreated youth compared to healthy controls. And she only looked at studies where they used emotional faces. And she found that kids who grew up being maltreated have greater amygdala activation. There are other structures also that she looked at, but just for the purposes of this talk, I'm going to just focus on the amygdala. Individuals who were exposed growing up to maltreatment, they have greater amygdala activation relative to controls. But the one issue here, and what we're trying to address, and I'll be talking about today, is that we were not able to examine specific types of maltreatment. So what most of these studies would do is they would take all kinds of maltreatment, such as being exposed to neglect growing up or being exposed to physical abuse, and just putting that in as one group and comparing that one group to kids who did not experience anything like that. So there's really a lack of specificity here. But still, she found the increased amygdala activation. Okay. Okay, but I'm not only interested in the amygdala. I realize that the rest of the brain is highly involved too, and really the field has moved to the idea that we have to look at circuits and interactions of different structures. So let me bring in some other structures as well, in particular structures of the PFC, or the prefrontal cortex. There's this idea out there, this theory that the prefrontal cortex may be involved in modulating or regulating the amygdala. And to support that idea, there's monkey work from Helen Barbas's group, a lot of it, this is one example, showing there's deep, dense connections between the prefrontal cortex and many areas regions of the amygdala and many areas of the prefrontal cortex. And this is work, and I'm going to talk about this more in a bit, that Lee Gotchis from my lab has undertaken, which I think is just really exciting. So using this measure, again, I'll talk about more in a little while, called it's diffusion MRI, and she uses an analytical technique called probabilistic tractography, where she mapped the white matter tracks between the amygdala and various structures of the prefrontal cortex, and she found really widespread connections between all these areas of the prefrontal cortex with the amygdala, but there's also variability, and some of the strongest connections in this case were with Brodmann's area 47, which is located right here, just over the temporal lobe. And also, so that had some of the strongest, the greatest connections with the amygdala, this area, and also the greater the connectivity on an individual level between the amygdala and the Brodmann's area 47, so those data were from diffusion MRI, that related to amygdala activation from functional MRI negatively. So the greater the connectivity between the amygdala and Brodmann's area 47, the less activation there was to threatening stimuli, threatening faces in the amygdala, suggesting that perhaps it's just correlational, of course, but suggesting that this area, Brodmann's area 47, may be involved in regulating amygdala activation. Okay, so that's threat circuitry. Let's talk about reward circuitry now, in particular the nucleus accumbens, which is located right here. It's on both sides of the brain, another structure deep inside the brain. So the nucleus accumbens is responsive, highly responsive to rewarding stimuli, such as winning money if you have them do a gambling task in the MRI, winning money, or also intrinsically rewarding stimuli, such as happy faces. And Jamie Hansen and colleagues, when he was at Duke, showed that teens who suffered neglect growing up, that's emotional neglect, have less nucleus accumbens activation to monetary rewards. And then turning to psychopathology, it's been shown that individuals, both kids and adults with depression or who are familial risk for depression, have less activation in the nucleus accumbens to positive signals such as happy faces. So neglect may relate to less activation, or seems to relate to less activation in the nucleus accumbens, and if that reduced activation appears to relate to higher incidence of depression. Okay, so let me just put this all together in a theoretical model for you. Again, this was pre-registered so we were not, to avoid cherry picking post hoc. So this is a theoretical model. So poverty increases the risk of two forms. There are many forms of adversity that increases risk for, but I'm going to focus on two. To violence exposure, such as abuse in the home and abuse and violence in the neighborhood. And it also increases the risk for social deprivation, such as neglect and experiencing less social interaction in the home as well. Just want to acknowledge that these two are related, but they can be statistically disentangled from one another. Violence also then in turn, in terms of the brain function, increases the risk or increases activation in the amygdala in terms of threat circuitry. And if you have more social deprivation, that alters reward circuitry, in particular relates to less ventral striatal or specifically nucleus accumbens activation. And then if you have these alterations in amygdala activation, you're going to be at increased risk for anxiety. If you have the altered, reduced nucleus accumbens activation, you're going to be at risk for depression. Okay. And I just want to acknowledge, I don't mean to be too dogmatic here. These two do have some crosstalk, but the prediction is, the a priori prediction is that altered threat circuitry will differentially affect anxiety, and altered reward circuitry will differentially affect depression. Okay, great. Oh, yes, sir.
Just a little question. Why is the PFC not in here?
Well, it is, but I didn't talk about it much because I have some data on that, but yeah, it's right here, but I didn't mention it. You're right. We're predicting altered ventral medial prefrontal cortex.
Thanks.
But yes, I didn't mention that. Thanks. Okay, so let's go to the data then. So these are the, these are the demographics. So the sample size, as I mentioned, we have collected data from 237 families. We have good neuroimaging data, depending on the procedure, from 165, between 165 and 183. This is like, we recruited them regardless if they participated in the scanner. So some of them had braces, some of them said, I'll go to the university, but I'm not going to go in that scanner, and we still brought them in regardless, because there's obviously really important to get other data from them as well. Okay, so they were between 15 and 17 during this visit, and it's a predominantly African American sample. Okay, so this is the annual income that was reported in this sample. You'll see a fairly broad distribution of income, but it definitely skews to lower SES. In fact, 43% of these families were at or below the poverty line of $24,250 for a family of 4. So it's obviously really, really struggling. And the vast majority of these families were below 200% of the poverty, and that's considered really economically challenged. So this is a lot of families are really struggling here. And the other point to make is that for me, there's a very different sample to be looking at, and very different for neuroimaging to look at as well. Okay, so this is definitely the saddest slide I'll be showing. So these are national rates of abuse that Katie McLaughlin found in a 2012 paper, and this is just to give you some context related to the reports of maltreatment that we see in these families from the Fragile Families Study. So across the board, the rates of maltreatment are much higher in this sample than the national averages. And actually, so 46% of this sample reported experiencing some form of clinically significant maltreatment. Again, much, much higher than the national averages for maltreatment. Okay, and all right, so this is what we saw. We not only interviewed the teenager, did case ads on the teen, and I'll present those data in a moment, but also the clinicians did a skid on the moms. It's a great opportunity to really meet the moms and talk to them and find out what they're experiencing. These are national rates for lifetime forms of psychopathology, and this is what we saw in this sample. So in a few areas it's elevated. It's not astoundingly high, especially considering the challenges that these moms are undergoing. But the rates for major depressive disorder and MDD, major depressive disorder, and PTSD, post-traumatic stress disorder, are roughly twice as high in this sample for lifetime relative to the national averages. Interestingly, social phobia and GAD, or generalized anxiety disorder, are actually lower in this sample. So the families, the moms are struggling, but they're not being totally wiped out here. They're doing okay. All right. Okay, so now let's turn to the adolescents. So based on the fact that these kids are growing up in high rates of poverty, high rates of maltreatment, and the moms have high rates of psychopathology, or twice the rate, at least for PTSD and MDD, I would think that the rates in these kids, these teens between 15 to 17 years of age, they will be elevated as well. And that's actually not what we see. So these are national rates for major depressive disorder, PTSD, and so on, and this is what we see in these teenagers between 15 and 17. So MDD is about the same in this sample as the national averages, and PTSD and GAD are actually lower. One thing just to highlight, I know a couple people are particularly interested in ADHD here. I am not an expert on ADHD, although I think it's really interesting and important, but we do see higher rates of ADHD, so we really have to, I have to learn about ADHD and figure out what may be going on there. But But yeah, so moving forward, this is really diverges a bit from hypotheses. We thought that the rates of anxiety and depression would be higher here, and they're really not. So we really have to, going forward, really try to understand what's going on with this, why we see this resilience, which is wonderful to see, but what are the mechanisms that may be accounting for this? Okay. Okay, so let me turn to the fMRI now. I'm not going to dwell on the biophysics of the fMRI acquisition, just that we used a 3T and we used a reverse spiral sequence that allows for better coverage of the amygdala, because that's one of the main targets of interest here. Mm. No. No.
On what percentage of the kids that you tried to enroll enrolled, and what differences there might have been between those? I mean, clearly,
Yeah.
one thing that might explain this is the ability of families to actually come an hour and a
Right.
half or 2 hours, and their willingness to come, both their
Right.
ability and their willingness. So did you, are
Right.
you worried about a selection bias, or is there
Right,
one?
so that's something, yeah, so that's something we're looking at. So there, for Toledo, and it gets a little complicated when we bring in Chicago, because we only were trying to get a subset of them. I think we only tried for like 40 of them and got a good number of them. But for just to give you some numbers, so the total number of teenagers from the original Detroit and Toledo sample, so from birth, so there are certain kids who've never been followed up with, but at birth they started with 425, and then we got 237 of those. And we have looked at the overall, like all the kids in these samples that that we got relative to all the available kids, and we don't see many statistical differences. I'm trying to remember. I think there's a slight difference in income, but not much at all. But I agree, that'd be worth looking at. We may be looking at the best off kids, yet they're still really challenged. But the nice thing about this sample is we know who the full sample is. We know who this representative sample is, so we could then statistically compare them, but we haven't seen a lot of differences yet. And one main reason for that is, for better or for worse, we pay them quite well. So if they want money, this is a good way for them to get money. So we have had people be very responsive, although they were really difficult circumstances coming to the university. But I agree, that's something that we definitely want to look at more. But our initial analyses were we did not see many differences at all between the full sample and our subgroup. Yeah. Okay, other questions? Okay. All right. All right, so yeah, I'll just go through, go on to the next slide then. Okay, so more with fMRI. So we did an emotional faces task. They saw to activate also threat circuitry, but also activate reward circuitry by looking at the happy faces. And they just show you what the subjects would see. So this would appear at the right in front of the subject. They'd see this fixation cross to indicate that there's going to be a face coming up. They'd see a face very briefly. This is a neutral face. Sorry, it's very small. A neutral face being presented for 250 milliseconds. Then we wanted to be sure that the subjects were actually engaged in the task and doing it. It was kind of early in the morning. Subjects were tired. I've fallen asleep many a time in the scanner. We want to be sure that they're not doing that. So we have them press a button indicating the gender of the face. Then moving on, here's a sad face being presented. Again, they press the button indicating the gender, and so on and so forth. There's a happy face being presented. So this task took 9 minutes. And subjects would see sad, happy, fearful, angry, and neutral faces. And 50% of the faces were models who were African American, and 50% were Caucasian. And we were particularly interested, I'm going to show you why in a moment, we were particularly interested in amygdala activation, but also, and what I'll be presenting here, is amygdala habituation, so how activation changes over repetitions, and also nucleus accumbens activation. Okay, so let me show you what I mean by habituation. I'm sorry, the screen's moving a little bit. I hope that's not annoying. All right. Okay, so this is my lovely daughter giving me this annoyed, disgust look that 15-year-olds seem to do. So the first time you might see this expression, you might show a lot of a large amygdala response. It's a very salient stimulus, very novel stimulus in the environment. So you give a really good amygdala response the first time you see it. But if you're like me, you see it a lot. And so through repetition, fortunately, the amygdala habituates, and so you just don't see it. It doesn't stay active for long. So this is what I mean by amygdala habituation. And so the reason why we did that, there are two reasons. One is that you can use the same category of stimuli as the comparison here. So with fMRI, there are no absolute values. You have to have a comparison stimulus, and that gets very tricky in terms of like if there are teeth in there or not, and how bright the face is. And with habituation, you don't have to worry about that because you're looking at the response to angry faces at the first half of the presentation, and then the response to angry faces at the second half of the task, I should say. It's also been found, this is work by Dylan G, and there are others that have found this, that it's more reliable than activation. It gives you more, if you scan an individual at 2 different points in time, the habituation is more reliable than the activation. Activation amygdala is not very reliable, so anything that can make it more reliable, I'm all for. Okay, so let me talk about the environmental measures, and this is Tyler Hein. I mentioned her earlier. She did that really nice meta-analysis. This is a major part of her dissertation that she's defending in just a couple weeks from now. And so she looked at 2 different environmental dimensions from the data from the Fragile Families study, focusing on ages 3, 5, and 9. So, and on one dimension was safety versus violence exposure. I'll sometimes just abbreviate it by saying violence exposure. And the other dimension is the amount of social support that the families are giving, or the neighborhood's giving, versus social deprivation. And so I thought this was really clever on her part that she looked at it at these dimensions at 3 different levels. So she looked at the child level, so for violence, the amount of abuse that the child is experiencing. And then for social deprivation, she looked at the amount of neglect that the child's experiencing. And again, these are at ages 3, 5, and 9. And then for violence at the family level, she looked at how much intimate partner violence, unfortunately, that's going on in the house. And social deprivation level, she looked at, or social support, she looked at the degree of the partner support for the mom or lack of support that existed in the household. And then at the broader community level or neighborhood level, she looked at reports of community violence during these ages, and she looked at the degree of community support and cohesion that existed during ages 3, 5, and 9. And then so she created composite scores from these dimensions, and then related it in a multiple regression analysis to amygdala function in the teen brain and nucleus accumbens activation as well. Okay, so all the results I'll be reporting there here are, we looked at current self-reportive stress, and the results still held using a measure called the AILS. And so the goal here is really to try to isolate, I'm not saying we did, but the goal was to isolate as best we can these variables from ages 3, 5, and 9. So we wanted to try to covary out the current teen's situation. And then also the results do not relate, for better or for worse, to anxiety and depression symptoms. All right. Okay, so this is a little bit complicated, and I will break it down, though. So This is the first brain imaging data I wanted to show. So looking at prior violence exposure and current amygdala function. So looking at the effects of violence exposure at 3, 5, and 9, while simultaneously, in a multiple regression analysis, simultaneously controlling for social deprivation and the interaction. So looking at violence exposure on habituation at age 15, we see a significant effect specifically on the amygdala. And this activation did not relate to anxiety and depression. But let me just break this down in terms of habituation, because it's kind of confusing for me to get my head around this, and probably for you as well. So this is the habituation we found. Let me first just show you the teens who experienced low levels of violence exposure growing up. They show a really nice habituation. So this is the first half of the faces. This is their amygdala response to angry faces. So the first half, they show a robust amygdala response. Then the second half, they show a lot less. So nice habituation there for those kids who didn't have a lot of violence exposure. But these are the kids who experienced a lot of violence exposure. So it's consistent and inconsistent. But the consistent part is that there's a lack of, we predicted there would be a lack of habituation in the amygdala in these kids to angry faces. And that's what we see. The thing that was surprising to us is the actual pattern. Overall, they're not showing a huge amount of amygdala activation. I would have predicted the activation would be up here and just would not habituate as much. But they stay low, but they just don't habituate. They just show this constant amygdala response, consistent amygdala response to the angry faces. So going forward, this may relate somehow to the relative resilience that we're seeing in these kids, and so we need to explore that further. Okay, so that's amygdala habituation. Let me show you what we saw with, in terms of reward circuitry, specifically looking at social deprivation at ages 3, 5, and 9, controlling for the amount of violence that they experience, and we see an effect on the nucleus accumbens. And what we found was the more social deprivation that they experienced at ages 3, 5, and 9, that relates to less nucleus accumbens activation in response to happy faces. And again, for better or for worse, these findings did not relate to the degree of anxiety or depression that they were experiencing. Okay, all right. So those are the fMRI findings, and I want to change gears. Are there any questions? Oh, yes.
Would you mind going back to the slide just before this?
Oh, sure, absolutely. The habituation slide?
Yes, please.
Okay. Yes.
I'm wondering, so when we look at the first half being less of a reaction to these stimuli, is this relative to some baseline? So is this, could it be that they were already activated, for example, by the whole scenario, and that they had less of a bump from this?
Right, I'm sorry, let me just clarify. So the first half, at least for these guys, they're showing more, correct? I'm not clear on what you're saying, like they're showing less activation in the first half.
So those with high violence exposure in their past seem to be showing less of a reaction in the first half
right
than
here.
Others.
Right here.
Am I reading that right?
Well, yeah. So just in terms of the figure here, this is not significant. I just want to highlight that. It's like there's no difference between the first half and the second half. But your point is well taken that in terms of and I didn't dwell on this, I should have, so breaking it down just to show you, this is looking at angry faces versus just a baseline condition and this baseline occurs throughout the task. So the idea if they're stressed, the idea is it should be factored out if these kids are more stressed. Does that answer your question?
Yeah, I guess I was looking at the difference not between the first and the second half, but between the first half in the one group versus the first half in the other group.
Oh, I'm sorry. I see what you're saying.
So maybe their baseline may have been different compared to the baseline in the low exposure group.
Right, but still the activation, we always have to do subtraction here, so we're subtracting out the baseline that it appears throughout, but yeah, it's never going to be perfect, so you're right, there could be a difference. I see what you're saying. You're talking about this versus that. Yes, yeah.
the same
Oh.
thought, and I'll
Oh.
pass it to you after.
great.
And was appreciating that you went back right here. I was going to save it to the end, but
Oh, sure.
it sort of made sense intuitively thinking about these people who have had chronic high violence exposure that maybe they aren't as sensitive, like their amygdala has sort of shut down in a way, and that would be why you're seeing the difference between the low and the high exposures in the first half.
Yeah.
Not even that, like maybe even the circuit is broken
Right.
and by the chronicity of the stress.
Right.
I don't know if that's helpful.
No, that definitely could be helping to explain what's going on here. Absolutely.
And just following on
Yeah.
with that, is there any data from wherever on that kind of chronic habituation? If you do this over and over and over again with somebody over multiple trials, do you get chronic habituation?
So let me see if I understand. Like with this group where we don't see the habituation.
Right, the hypothesis would be these guys have been habituated over their lives.
Yeah, absolutely. Right, and so this is specifically in response to angry faces, and so going along with that is perhaps they're seeing angry faces all the time, and they're just used to it, and so they're not going to respond. But yeah, and the hypothesis would be though that maybe over time they will habituate. It's just going to show a different pattern, and we just don't have the data beyond this second half, unfortunately.
Two microphones.
I know that you're not necessarily presenting on any of the data from the HPA access. And I'm just curious if that's been looked at all in terms of this chronic habituation with HPA access and cortisol level.
It has been looked at. Yeah. I'm not comfortable like going into detail about it,
Sure.
but yes, it has been looked at, and the findings are. She looked at the same. This is Melissa Peckins, a postdoc. She looked at the same thing of the violence exposure and deprivation, and overall, there's in response to a physiological challenge of putting your hands in a bucket of ice water, which is a
Like cold
standard
presser
procedure.
test, right?
Exactly, the cold presser. That they show a blunted response, and yeah.
Thank you.
Okay. Oh yeah, sir.
Well, I'm just thinking, has anyone fit any of this to the construct of callousness?
Yeah, so that's so Luke Hyde, and I'm going to acknowledge all the tremendous people who have worked on this. Luke Hyde is a collaborator of mine, and my interest is in internalizing, his interest is in externalizing psychopathy, callous unemotional traits, and that's something they are looking at as well. And that I'm not in the position to say what the findings are. I just don't know. But yes, that's a real interest as well. The grant is on internalizing because it's through NIMH. They're not interested in funding callousness, but that's another whole area of interest. Yes. Was there another question or? All right, I have more slides, but. All right, great. So let me just plow ahead. I've been spending a lot of time talking, so that's good, but I'm running out of time. So let me just quickly talk about diffusion MRI, which I'm really excited about. This is a new area to me, but just in the interest of time, I'm very briefly going to describe what's looked at here. So diffusion MRI allows one to look at white matter tracks by measuring the movement of water inside the brain. And so there's a lot of water obviously in the brain, and there's a lot of room for not only this way but up and down, that's called isotropic, or it allows for diffuse movement. But when you have a lot of white matter, that's going to constrain the movement of these water molecules, and in that case you have anisotropic, or very focused movement. And what diffusion MRI measures the degree of how much focused movement there is, with the idea that is those are white matter tracks that it's indexing. So that's diffusion MRI, and what Lee Gotches, as I was mentioning earlier, did, she used diffusion MRI to analyze the data with something called probabilistic tractography that allows one to quantify the probability of connectivity between a region and every voxel of the brain. And So these are data I presented briefly at the top of this talk, but just to remind you, so with diffusion MRI, you can map the white matter tracks from the amygdala to all the structures of the prefrontal cortex and see which has greater connectivity than others. Again, she found that Brodmann's area 47 had particularly strong connectivity, and the degree of connectivity she found, and this is in this sample, related with Brodmann's area 47 related to amygdala activation. So the idea is you have stronger connectivity between Brodmann's area 47 and the amygdala. That may help you to regulate your amygdala response better and perhaps protect you from things like anxiety disorders. So in addition, Lee is very interested in looking at how these types of environments, how the degree of violence exposure and degree of social deprivation, how that relates to white matter track development using probabilistic tractography. So she looked at these dimensions, the degree of safety to violence exposure, the degree of social support to social deprivation, all these at ages 3, 5, and 9, and then also the interaction of these two. And let me just tell you that she did not find, I find this so surprising, she did not find a direct effect of violence exposure on white matter development between the amygdala and prefrontal cortex. She did not find an effect of social support and white matter tract development. But what she did find is she found an interaction between these two and the white matter. And let me illustrate this for you. So, this is part of the interaction. I'll show you the other part in a moment. So this is the white matter tract connectivity between Brodmann's area 47 and the amygdala, and the degree of violence exposure at 3, 5, and 9. And this is just looking at kids who experienced a lot of social deprivation growing up. In those kids, the more violence exposure they have, those kids show less white matter connectivity between the amygdala and the prefrontal cortex, but just those kids. What we see in the kids who experienced a lot of social support growing up, they don't show that pattern. And let me just back up. This is a significant slope. This is a significant slope. Oops, sorry. This is not. So I just want to qualify that. But this is a significant interaction between the connectivity between the amygdala and the prefrontal cortex. So these kids who experience a lot of social support, in the face of more and more violence at ages 3, 5, and 9, they're not showing that weakening of connectivity. So the idea is perhaps this social support is somehow buffering these kids, and in turn, the greater white matter connectivity is buffering that amygdala response. Okay, so I'm going to come back to this in a moment. I find this complicated, and I'm just trying to wrap my head around it. Hopefully I explained that. Okay. All right, so this is, I just want to, in summary.
A question.
Oh, sorry. Oh, sorry, same age. Thanks for bringing that up. Yeah, so these are at 15 to 17, the same scan. Thanks for clarifying that. So this is, again, just as with Tyler's, this is ages 3, 5, and 9, exposure to violence and exposure to social deprivation, and then the scan is done at between 15 and 17 years. Okay. All right, so just to wrap it up, so this is, again, just our theoretical model. I already walked through this, but just to remind you what we pre-registered, and this is what we found. So we did find that more violence exposure relates to amygdala activation. We are currently looking at functional connectivity, which I'm not going to go into, between the amygdala and ventral medial prefrontal cortex, but we did not find these things relate to anxiety disorders. Again, the rates of or symptoms. The rates of anxiety disorders are really quite low so far in this sample, so maybe that's why we're not picking this up, but we're not even seeing it at symptoms yet. Same thing with social deprivation, degree of social deprivation related to less nucleus accumbens activation at between 15 and 17, but that did not relate to depression disorders or depression symptoms yet. Okay. Oh, yes.
So did you differentiate boys and girls?
We've
You would expect really different responses for boys and girls.
we've explored. We broke up gender, looked at males and females, and it doesn't explain. It's not driven by one sex or the other, right? But I think that's a really important point because in regular usual sample of adolescents, the depression rates are going to be much higher, but we're not seeing, it doesn't explain the data. We do, in the brain imaging data, I should say, we do covary for gender, and that cleans up the data, but yeah. Okay. All right, so let me just give you a more specified model based on our data that is in progress, and for now I'm really just focusing on the violence exposure piece of this. So poverty increases the risk for being exposed to violence in childhood. Then in adolescence, we found that it altered threat circuitry in the amygdala. We're working really hard to be able to follow these youth into adulthood. We followed up with them, as I said before, 2 years after the scan, but we'd love to continue to follow them through their transition in adulthood. And so we'd be really excited to see if this, maybe we don't see anxiety and depression yet, but it may come up. They may start to have the rates more consistent with the moms, and so it may relate to how they do later on in adulthood. Okay, and then bringing in the new data. The blue is the new data. So we found that increasing social support related to greater Brahman's area 47 right here, and amygdala white matter connectivity. Oops, sorry. And that in turn tamps down the amygdala response. So social support and this white matter connectivity may be a buffer for amygdala activation and may also in turn buffer the risk for anxiety and depression. Okay, so it's 9 o'clock now. I have this little thing on reproducibility and replicability. Maybe I'll just skip that. I'm happy to talk about that.
Go for it.
Oh, okay, great. Okay, so in psychology and more in neuroscience, and I think it's sweeping the world of research, reproducibility has become a really big issue, and I'm really trying. I've got to probably do research for another 20 years or so, and I really would like to have that research be solid findings that will last. And so we've taken this very seriously, and so we made sure that our sample size was big enough for the questions that we're asking. It's not an ABCD study, which is just monstrous, but for existing fMRI, particularly with these longitudinal data, it's a large sample size. Also, as I was mentioning, we pre-registered our hypotheses, and we will continue to do that before looking at additional data, so that it's clear that we're not just, we have, obviously there are thousands and thousands of variables. We could create wonderful stories about them all post-hoc, but we want to be clear that that's not what we're doing, and we're testing these things, testing hypotheses that we came up with a priori. We have a priori regions of interest and whole brain corrected analyses. This is derived from a representative sample, so we know who this sample generalizes to. Often people will recruit via Facebook and other things, which is fine for some things, but you don't know who that sample is. So these findings, other people may not be able to reproduce them if they're looking at, say college students or something like that, but if they're looking at a similar sample, we're saying that they should be able to reproduce. And also, finally, the data are being shared on an NIMH RDoc server, and that's going on now and is shared. And then we will share our code so people can look to see if there are problems beforehand. And okay, that's it. And so I just want to really, this is the most important slide, acknowledge all the tremendous people who have worked on this project. This is Coulter Mitchell. He's a geneticist and sociologist. Nestor Lopez Durham and Luke Hyde. I mentioned earlier, they're the clinical psychologists on this, but Nestor does tremendous work on HPA axis function. Luke, as I mentioned, does externalizing. Sarah McClanahan's the PI of the overall Fragile Families study. And these are all the tremendous graduate students and staff who went to Detroit, went to Toledo, picked up these families. I want to make a special mention of Jaime Munoz Velasquez, Adrian Woods, Kristen Stauffer, who else? Ariana Gard, and I did mention earlier Tyler Hine and Lee Gotchis and with their data. And that is it. Thank you very much.
Dr. Monk, to follow up on that last point,
Yes.
where do you pre-register? Is that on like clinicaltrials.gov, or how do you, what's that process?
Yeah, that's a good question. I think there are options. I know of, but I'm not used clinicaltrials.gov, but we do it through a University of Virginia open science website, and it's very easy, and you just kind of do it yourself, and it's not like it needs to go through this external approval process. You're just putting this out there saying, this is the date, you have a timestamp, this is what we predicted. And actually for Tyler's, we made a change to it, and it always had bothered me, like, well, if you pre-register something, you're really kind of stuck with it, for better or for worse. And she was talking to a colleague, Adrienne Belt, who's this tremendous, she's a psychologist and a biostatistician, and she suggested, well, you should also look at the interaction. And she said, yeah, we should look at the interaction too separately. And so she then revised her the pre-registration, and that's all documented. And so yeah, no, it's been a really good experience, and you just see like, oh yeah, they made this change to it, and you can make those changes. But yeah, it's through UVA. I don't have the exact website off the top of my head, but yeah, it's been tremendous.
Thank you so much for a wonderful talk.
Yeah.
I was curious, given that you have these families, part of the Fragile Family Study, I was wondering, do you know if any of them have received any form of intervention? And do you have any thoughts on how that might have complicated your data or your findings? Families who have either been involved in child protective services, or kids who have received access to charter school educations, or even psychotherapy?
Yeah, so that's a good question. We know there are a few who've been notified through Child Protective Services, and in fact, we've had to actually contact Child Protective Services on a couple of occasions for a couple families. But yeah, so that's we have general history questions that we ask about. We have not seen much. Any kind of psychotherapy that I know of. Very little psychotropic meds are given to these kids. There's few of them have ADHD meds, but not a lot. But that'd be something to look at further using the history. The other thing is you're bringing up charter schools. So the largest sample comes from Detroit, and as you may or may not know, it's become this horrible testing place for-profit charters. And so there are a lot of these families that are in charters, and they switch one to another as places open and close. And that's not something we've looked at. We do have those school records, as mentioning, and I think it'd be a tremendous opportunity to look at broad measures of schools and see how that relates to how these teens are doing now and into the future. But yeah, there are all these really important variables that you brought up that we just have not been able to, we have not looked at as of yet, but would be great too. Yeah, Sarah.
Thanks for sharing your research with us. It's certainly thought-provoking, and I had a few thoughts about the exposure side of it.
Mm-hmm.
Given your sample, I wonder about exposure to racism.
Yeah.
Hard to kind of get at the structural thing, but interpersonal racism, was that documented in this study?
That's a really important issue. So Jaime Munoz Velasquez, who I mentioned, has a real he's a graduate student with me, and that's what he wants to do and has been trying to do, is looking at discrimination in this sample focusing on African Americans. It's really almost all African American or white. There aren't many Latinos happen to be in this sample. There are in the Fragile Families, but they're in Texas and California primarily. But yeah, that's something we've looked at. And unfortunately, so Fragile Families, the overall study, unfortunately did not ask questions of that. So we do not have longitudinal data on discrimination, but we included a couple measures of it. And the surprising thing is that the self-reported rates of discrimination are really low. And we couldn't, and so I guess that's not that uncommon, working with a collaborator, Debbie Rivas-Drake, who does a lot of this kind of work. And she says that's not unusual, and she showed us some ways to statistically look at the data, but it didn't relate to symptoms, and it didn't relate to brain. And maybe one idea is that in Detroit, they're fairly protected right now. The majority of these kids who are African American are in Detroit. Perhaps they're not getting the face-to-face discrimination that they're reporting. They're certainly getting the structural discrimination, but that's not what the question asks. But so he's moving forward to look more at structural racism that leads to segregation, and looking at measures from census tract data, things like that goes structural racism information that we could then relate. But yeah, we were surprised that we're not getting high rates of self-report instances of discrimination. But going forward, as they become adults, we're definitely going to continue to look at that. Yeah.
Because the birth outcomes have been shown to have a pretty strong association with exposure to interpersonal racial discrimination. So
Yeah.
maybe it will come out
Yeah.
in some way.
Yeah, no, we really are interested in continuing to pursue that, and it's an excellent sample to do that with. Yeah.
I just had another question. Okay, just about exposure too. You've got a great big mega construct, it looks like, and I like the idea of having these multiple levels of, exposure to threat and exposure to support. But I wonder if there's a lot of noise with 3 levels, and whether if you sorted out the specific interpersonal, physical abuse from the neighborhood violence, and I don't know if you've done that differentiation.
Yeah, we've not done that. And so this kind of gets where we're a little hamstrung, and I don't have an answer to this. Back, a while ago, before these justified concerns about reproducibility came out, we would, you have to explore the data to some degree, but we'd explore it more than we do now. And we have not looked at that because we have to come up with a hypothesis and pre-register it and then look at it. There's just no room for really trying to, it's hard to really justify trying to understand the data until you have a pre-registered hypothesis. So we've not looked at it, but we are interested in taking some of these things and looking at more, what is it about, say, the social support, really drilling down on social support and trying to better understand that. So Lee Gatches wants to put together a pre-registered hypothesis focusing more on that, and then maybe also looking at, are there particular times in development when those supports are more important than other times, and that'd be great to look at as well. Yeah, and at this, with the Tyler's work and then Lee's current work, we've not been able to look at developmental timing at all. We just created these composite scores from 3, 5, and 9, just trying to generally, as a first pass, look at childhood overall. But yeah.
Thank you. I also want thank you for the great talk and how carefully that you're looking at these issues. I also wanted to ask about I know that you're interested in the issue of poverty and how that impacts, and I can see that in this sample you have both poverty and race that may be confounded, or at least partially confounded, and wondering what your thoughts are about how you're going to try to separate the impact of poverty.
Mm-hmm. So when you say poverty, I mean, I see poverty as relating to all these things. And for the first pass of the data, we are looking within this high poverty sample at these forms of adversity that are common in poverty. But when you say poverty, are you thinking specifically about the economic hardship?
I'm actually just interested in
Oh.
what you're thinking and,
Oh,
kind
yeah.
of how you're looking at poverty and what you're thinking about how that may be impacting your findings.
So this work, our first pass, trying to look at these salient variables of poverty, that's what we're doing. But then another area more specifically about the financial piece is we're interested in economic hardship. And the Fragile Families data, the longitudinal data is not good for discrimination, as we were just talking about, but it's really good for economic hardship because there are economists who helped start it. And so we're in one thing that Melissa Peckins and Jaime Munoz Velasquez are doing is specifically looking at measures of economic hardship and also change in economic conditions from between 3, 5, and 9, and how that impacts, how that affects brain function and psychopathology. So that's another area that we're moving forward with. But more broadly, I'm trying to move away from looking at poverty, just as a variable, just as poverty and as a, income or maternal education. I just don't find that very satisfying because it's so broad and there's so many things that are going on there. I'd really rather try to be more specific and take these other variables and treat them as covariates to help us pinpoint more specifically what conditions of poverty may be altering these measures of interest.
Thanks.
Yeah.
This is just would really help me with my general education. So you looked at exposures prepubertally and then looked at postpubertal brain stuff. Could you just talk a little bit more about sort of, trajectories of brain development or, sort of where plasticity happens and kind of, why or why not you didn't maybe look more at sort of what was going on in the current, the kid's current environment?
Yeah, well, I just say we have looked at the current environment as well, but we're particularly interested in just making use of the longitudinal data. So that's what we're publishing from. But we have looked at, current reports of maltreatment. They're somewhat similar to this with the kids' self-report. It's somewhat similar to what's reported at 3, 5, and 9. But yeah, so like just really, we're looking at the longitudinal piece because we want to make use of the great data set from the Fragile Families. But just broadly, there are thought to be 2 big times for plasticity, and one is in infancy, but then there's this other one, this hypothesis to come up that's more controversial, but it's thought to be around is right at the beginning of puberty, and there's a lot of the idea there, and this is work from Jay Geed and other people, that there's a lot of opportunity for change. You see a lot of synaptic connections happening right at the onset of puberty. I wish we could get more specific, and the last data that they have is from 9, but it'd be wonderful if we had more data sometime between 9 and 15 to get a better handle on that specificity and if it has anything to do with pubertal timing. I mean, one idea, yeah, it definitely could. But certainly the brain is, as we all know, the brain is plastic throughout life. There's huge opportunities for change, for better or for worse, but that may be the early puberty may be a real salient time when the brain is open to the environment. Yeah. Great. Oh, yes.
Hi, thank you for your talk. I have a question about your reward data.
Yeah.
I'm wondering if you've thought about happy faces as differentially rewarding in this sample compared to something like a monetary reward.
Well, let me just first say, I actually want to hear more about what you're thinking. That sounds interesting. But let me first say we also have a monetary reward task. I just didn't have, I did not have time to talk about, but actually we've not even fully analyzed the data in terms of social deprivation. But we use the monetary incentive delay, which is this gaming task where you win or lose points. We didn't want to use money a lot of people use money because that's going to be a really good reward, but we didn't use money because we were concerned that there was, there's going to be a difference driven by economic circumstances. So instead we use points, which still, even though it's just a game, it still drove subjects to do as well as they could. But maybe you could, with that in mind, maybe you could tell me more what you're suggesting with the differences in response to happy faces, so based on their environment and how they interpret the happy faces?
Yeah, that's exactly what I'm thinking.
Yeah.
If these children have been in an environment of maltreatment and potentially less opportunities to see happy faces, have they developed a different response to that over development?
Yeah, no, and that's kind of like, that's one, we can't determine if that's it, but that's one of the ideas, that the lack of exposure to these intrinsically rewarding stimuli, if that's shaping the brain, and that's what we're seeing here at age 15 to 17, that certainly could be it. Yeah, we have them after the fact identify the faces, because there's some concern that maybe a subgroup of these kids they don't recognize different emotions, and I know that they all do a good job on happy faces. Happy faces are very, very easy to identify. They're very different from other faces. So they do that correctly, but it'd be worth looking at differences in reaction time. Maybe they're a little slow. Maybe some kids who are going to be a little slower at correctly identifying happy faces. But yeah, that's worth seeing. Yeah.
Thanks.
I feel like we could continue this discussion longer, but we are out of time. And I do want to thank you for sharing this really rich data with us. It sounds like we may need to have you come back again another time to explore more future data analyses you're going to do. I do want to ask people first, let's just give another round of applause for
Thank you.
Dr. Monk.
Thank you.